Bladder cancer is the 10th most common type of cancer worldwide and is often linked to exposure to harmful chemicals, such as those found in tobacco smoke. A new study shows that the bacteria in our guts may play an important role in bladder cancer development. The scientists showed experimentally that certain gut bacteria can transform a class of carcinogens, often found in cigarette smoke, into related chemicals that accumulate in the bladder and give rise to tumors. At any given time, over 10 trillion microbes call our guts their home. From breaking down nutrients in our food to strengthening our immunity against pathogens, these microbes play an essential role in how we interact with the world. This includes, as shown in a new study by EMBL researchers and collaborators at the University of Split, Croatia, the way the body responds to carcinogens and develops cancer. Carcinogens are chemicals that can cause ordinary cells to transform into cancer cells, giving rise to tumors and cancer. They can be found in a number of places, tobacco smoke being one of the most well-known sources. Researchers have previously found that if mice are exposed to the nitrosamine BBN, one of the chemicals found in tobacco smoke, they reliably develop an aggressive form of bladder cancer. This is therefore used as a common laboratory model of carcinogen-induced cancer. Researchers studying this model when they made a curious observation. If the mice were fed antibiotics at a dose that kills 99.9% of their gut bacteria, at the same time as they were exposed to BBN, the chances of them forming tumors were much lower. While 90% of mice exposed to BBN went on to develop bladder tumors, only 10% of those that also received antibiotics did so. This led to hypotheses that the gut bacteria might be involved in regulating the way BBN is processed in the body. The decrease in tumor incidence was so dramatic that at first researchers doubted the results, thinking they must have made a mistake somewhere in the experiment. Consequently, they repeated the experiment 5 times before they finally became believers. It was fantastic to realize that with a treatment in this case, antibiotic scientists were able to abolish cancer development. Using a variety of microbiology and molecular biology methods, the researchers discovered that bacteria living in the mouse gut could convert BBN into BCPN. Like BBN, BCPN belongs to a class of compounds called nitrosamines. However, the team found that, unlike BBN, BCPN concentrates in the urinary bladder and triggers tumor formation in a microbiome-dependent manner. The researchers next studied over 500 isolated and cultured bacteria to identify the specific bacterial species involved in converting BBN to BCPN. They found 12 species that can carry out this carcinogen biotransformation. Researchers sequenced them and were surprised to find that many of those species were skin-associated and found at relatively low abundance in the gut. They speculated that there might be some transient transfer of such bacteria from the skin to the gut as a consequence of the animal's grooming. But it was important to figure out whether these findings would also be true for humans. Following these initial studies in mice, the scientists used human fecal samples to show that human gut bacteria can also convert BBN to BCPN. As a proof of concept, they showed that if human stool was transplanted into the intestine of a mouse that had no gut microbiome of its own, they could also convert BBN to BCPN. However, the researchers also observed large individual differences in the ability of the human gut microbiome to metabolize BBN, as well as in the bacterial species involved in the biotransformation. This lays the foundation for further research to see whether a person's gut microbiome represents a predisposition for chemically induced carcinogenesis and could hence be used to predict the individual risk and potentially prevent cancer development. This difference in interindividual microbiota could explain why some people, despite being exposed to potential carcinogens, do not develop cancers while others do. Do these findings mean antibiotics can universally prevent cancer? No, of course not. This calls for more studies, including some that we are doing currently, to understand how the microbiome influences the metabolism of different types of carcinogens. It is also important to remember that cancer is a multifactorial disease. There is rarely a single cause. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.